Really just to share with you the science that we are most excited about. Ken gave us very specific guidelines. He said, don't make it too simple. Keep it complicated. We want to know what's really happening in the lab. That's right. He said, these are very smart people. So that's the plan. And if it's too much, you can blame Ken. The plan is really to start by telling you a little bit more about the Institute, how it came to be, and what its purpose is, and then to kind of dig into the science, mostly focusing today on memory and on movement and how our brain accomplishes both of those things, and some of the open questions remaining that we're looking to solve in the next 5 to 10 years. Before I give you sort of more detailed information about the Institute, I'm excited to ask
thoughts on the future of the Zuckerman Mind Brain Behavior Institute. And what I'm going to tell you very briefly today is that the Institute, which addresses inordinately complex questions of mind, now recognizes a sense of responsibility, a palpable urgency to apply this insight to neurodegenerative disorders.
But I'm also telling them to you because I think many of us have reached the conclusion that that is not enough. We cannot just sit around and wait for curiosity to drive discovery and to change the world. It doesn't work as well as we need it to, and we can see many problems, pressing problems in society that we know will end up having scientific solutions, and we don't have the solutions that we need right now.
So I am going to tell you about my curiosity-driven research, but I think it couldn't have been put in a better context that one of the reasons that I am at ZI, which I have only come to in the last 2 years, is that it was the place that I really wanted to do this curiosity-driven research because the context and the goals of the Institute in how to shape this research into a deeper understanding of our own brains or even potential mechanisms for helping with health and disease issues is something that I couldn't have done in the place I came from or in a sort of more siloed research institute. So we're really excited to be at the Zuckerman. He really was the one who made Drosophila, this fruit fly, an organism we could study, because now that we saw how its genes worked, we could start to manipulate those genes for scientific research purposes to affect the animal. And fast forward it to the last decade, we now have tools in the fly where we can manipulate their genes to target specific brain cells, individual neurons, which means I can go into my fly and I can turn a single neuron on, I can turn a single neuron off, or I can express fluorescent proteins in that neuron so that I can watch it blink on and off when the fly is behaving. And these are the components that you need to go in and be able to understand a system as complex as the brain.
The things that's clear about both Alzheimer's and other neurodegenerative diseases is that there is something that's starting within cells. So it's starting at a cellular level, and then as Gwyneth very elegantly described earlier, those cells and circuits starts to then kind of have a cascade of an effect, where it's almost like a set of dominoes. So one cell goes bad, that cell is connected to many other cells, they start to go bad, and so it starts to almost in a way spread throughout the brain, and that manifests in the sort of gradual impairment that we see in people with Alzheimer's and other horrible neurodegenerative diseases. And so really what my lab is doing at Zuckerman is to really focus on the subcellular. So Gwyneth had mentioned the connectome, which is how different neurons and other cell types in the brain connect to each other. We are going right into the very heart of single cells and trying to understand what is that first event that actually tips this whole thing off into a kind of cascade. And we've had hints about that for a very long time. Ken mentioned Alzheimer. So one of the things that Alzheimer found was that when he looked at the post-mortem brain of people with the disease that now has become named after him, he found that the neurons were kind of clogged up with very clear globs of kind of gunk. And that sort of showed very clearly that the neurons were filled in a way that healthy neurons from someone who died of a heart attack or something else did not have in their brain. And to Richard's point earlier about, neurons and your other brain cells have to last a lifetime, which can be up to 100 years or more, because they don't divide, you have to think that all of the protein molecules that are inside each individual brain cell, they're all performing very important tasks to the maintenance and upkeep of your cell, and that for those to survive 100 years or more, they get recycled all the time, but sometimes those protein molecules, which have each got individual functions, and those functions are defined by their three-dimensional shape, sometimes they don't adopt that shape, and they sometimes start to accumulate in ways that cause this gunking up of the cell.